Okay, so in this lecture, we are starting your unit 10, Microbial Nutrition and Culture. So we already study about how microorganisms grow, and basically in this lecture, we had to see what kind of thing which are going to responsible or which are going to help microorganisms grow, and what are their cultural practices. So, bacterial growth is proliferation of bacterium into two daughter cells. So, basically, when they grow or they divide and multiply, so mostly the process which is involved in their growth or in their division, that is the binary fission. And you all know how the binary fission happens. This is the same kind like the cell division happen. First, the karyokinesis and after the cytokinesis, and one cell become in the two cells. So this is a very normal kind of process which happen inside the bacterial growth. But the major thing what we have to study the type or their growth or the factors which are affecting the bacterial growth. So, providing no events occur, the resulting daughter cells are genetically identical to the original cell because this is a kind of mitotic division. This is not the meiotic division. So, mitosis, we have the identical copy of their parents in form of the genetic material. So, because genetic material do not go under the reduction division, so hence we have the same kind of genetic material as their parent have. So both daughter cell from the division do not necessarily survive. Both daughter cell, which has been made from the division, do not necessarily survive. Means they can be or can be not. Sometimes, if the conditions are so favorable, nutrient amount is enough in the quantity, and both the have potential, so they can be survived or to their next generation. But if there is anything in lacking, so we are going to see what are the factors which can be useful inside their whole survival. So, if the number surviving exceed unity or average, the bacterial population undergoes exponential growth. So, this term exponential growth, we were going to see it when we are talking about the analytical phase of bacterial growth, means in the numbers. Like this is some kind of predicted value. We can predict the bacterial growth by seeing their first or second generation because we know the pattern in which they are going to grow. So first we are going to see what are the microbial growth requirements. So the first topic we are going to discuss here, that is how microorganisms grow and what are the basic requirements of the growing of microorganisms. So you can see there are two type of requirement we need here. First is a physical requirement and second one is a chemical requirement. So in the physical base, we have these all the physical aspect, all the abiotic things here: temperature, pH, oxygen, hydrostatic pressure, and osmotic pressure. So these are the things which are not going to make any kind of chemical change inside the bacteria or inside the microorganism. So that's why we call them the physical requirement. And the chemical requirement, on the basis of chemical requirement, all the biochemistry of particular microorganism community deal with, like the carbon compound, or you can say the carbon amount, nitrogen, sulfur, phosphorus, trace elements, other micro elements which are needed for their growth, oxygen, organic growth factor. So there are two categories in the requirement for the growth of microorganism or bacteria: physical and chemical. So you have to remember these, that we need two kind of requirements: physical requirements, which going to include all these things, and chemical requirement, which are going to add on these all chemical perspectives. Now we are going to see the first physical factor that is required in microorganism growth, that is pH. So you all know pH is required in everything, like inside our body, and if any kind of experiment we perform, which is based chemical based, any kind of chemical based practical, so we need to be consider the pH has to be accurate according to need of experiment. So same here, pH is also very specific kind if we wanted to grow the microorganism, and it is also specific to the microorganism community. Like all community do not grow on the same pH. They have a specific pH to grow. Like most of the organism, they grow between the pH 6.5 to 7.5. That is called like optimum pH. The pH at which the microorganism grow best is like pH 7, or even for our body, this is the optimum pH. We also need the pH 7. The neutral phase, neither acidic nor basic. So the molds and yeast grow between the pH 5 to 6. So you can see they are slight acidic because they are growing to the down, or you can say to the phase in which pH have some acidic mode. According to their tolerance for acidity and alkalinity, bacteria can be classified in the three groups. So how they can be grow on what kind of pH? On that basis, we did these three kind of classification for on the basis of their pH tolerance. First is here acidophiles. So as the name suggesting, they are very fond for the acids, or they have loving nature towards the acid. Like we talked about the hydrophils, the thing which are favorable inside the water. So same is here acidophiles. So they actually love to live inside the acid. Grow best at pH 0.1 to 5.4. So basically, their growth pattern, like how and at what pH they are going to grow best, that is a pH 0.1 to 5.4. Second one is here the neutrophils. So neutrophils is like you can see they are almost neutralized, neither acidic nor much basic. So they are going to grow best at the pH 5.4 to 8. This condition, neither it is too acidic and neither it is too alkaline. So alkalinophils, so they are going to be base loving because as their name suggesting, and they have to be grow best at the pH 7 to 11.5. So these are the certain conditions in which bacteria are going to grow on a certain pH. So just remember these three terms: acidophile, neutrophiles, and alkalinophiles, and they are all based on the just the pH number. Now, the second physical factor which come here for required for the growth of microorganism, that is temperature. So according to temperature also we have some categories in which we can put the microorganism, like in the acid we put them into the three categories, but on the basis of temperature we are going to put them into the four categories. So first category here you can see the psychophils. So the psychophils, they are, you can see they can grow best in between the 15 to 20 degrees Celsius. They are the low temperature organism, or which can be just survive at a very low temperature, even below to the room temperature. Because the optimum room temperature we think about is like 30 to 37 degrees Celsius. But they are the psychophils which can grow just below the room temperature. Second one is cyclotrophs. So they can be grow between the 0 degrees Celsius to 20 to 30 degrees Celsius. Their range is pretty big. Like we can, some of the cyclophils also come under the cyclotrophs. So basically, this is between the 20 to 30 degrees Celsius. Third one here, the mesophiles. So mesophiles can grow in between the 25 to 40 degrees Celsius. And lastly, the thermophiles, they can grow at the higher temperature like 50 to 60 degrees Celsius. We are talking about here just like the EU bacteria. Okay, we are not talking about here the archibacteria, which can grow in a very extreme conditions like the 100 degrees Celsius, 120, 150, 160. They are the exception, and that's why we put them into different domains. So that we are talking about the bacteria which we can culture inside the lab, or which we can do how their cultural practices work. So that's why we just take these four, you can say the four conditions or four terms for temperature-based bacteria. Typical growth rate and temperature if we see, so there are three typical growth rates which actually we can use for culturing the bacteria. First is a minimum growth temperature, so lowest temperature which the species can grow. So these can be the three growth rate which we can apply on the bacteria and see how they can react to these specific kind of temperatures. So minimum growth rate, we just give them a very minimum and lowest kind of temperature. Optimum growth temperature, we give them a moderate kind of temperature, neither too hot and not too cold. And maximum growth temperature means the highest temperature at which they can possibly grow. So actually, we are seeing their survivability and tolerance actually, how and what temperature they can tolerate and grow. So on the basis of these, we can actually doing the research, and we can see that at this temperature they are growing best. This other temperature they are having moderate growths, and the third temperature they cannot grow at all. So on the basis of this typical growth rates and temperature is just to perform to see how microorganisms react to the specific kind of temperature. Next, you can see a graph they plotted here just to see the rate of growth or on the basis of a specific kind of temperature. So the best kind of growth is, specified to the specific kind of organism. Like the psychrophils, they just grow in between 10 to 20. Psychotrophs between 20 to 30. Mesophiles can be between 10 to 50, or mostly at the 40. If we see the graph is like this, 40 to 70 in between, they grow best at 60. In between the 70 to the 110, they grow best at the almost 95 or 92 around. So that is a actual growth rate curve we can make on the basis of their rate of growth and the temperature on which that specific rate of growth we obtain. Now, the food preservation temperature. So, as we do the food preservation by heating it up, like like if we wanted to preserve our milk, we just have to uh, heat up like on approximately 3 to 4 minutes on their boiling point. So, temperature in this range. What why we are doing that, to destroy the microorganism which can make the contamination inside the milk or any kind of food material. So temperature in the range destroy most of the microorganism, although lower temperature take more time. So if we do the boiling at this temperature, most of the microorganism, most of the bacteria is just going to be destroyed. In if we are talking about like the temperature, they are just little below, like slow bacterial growth at this temperature. They will take more time to kill the bacteria. Same here. For as we just no significant growth below the freezing also. So that depends what kind of temperature we can give them and how their microbial growth depend on that. This is not okay for all the kind of microorganisms, that is also specified to each other. Now, the oxygen, that is a third physical factor which is responsible for the microbial growth, and on the basis of oxygen consumption, we divide them into the several groups. Like the aerobes, which require oxygen to grow. So, not all the bacteria, or either they can be required the oxygen for their metabolic activities, or sometimes they do not require oxygen at all. So on the basis of their requirement, aerobes, they require oxygen to grow. Obligate aerobes must have free oxygen for aerobic respiration. So they also obligate aerobes, they use the free oxygen for their aerobic respiration, pseudomonas. But not the condition. Sometimes they can also do their oxygen inhibit zones, but for their aerobic respiration, they need it. That's why we call them the obligate. Anaerobes do not require oxygen to grow at all. They never require in any phase. Obligate anaerobe killed by the free oxygen. So they are just opposite to the aerobes. Like if we expose them to the oxygen, or if they mistakenly come into contact with the oxygen, they are going to be killed. Like the bacteroids, they are the very specific and best example of obligate anaerobes. Next one is here microaerophils. So microaerophils, they can grow best in presence of small amount of free oxygen, not bounded oxygen, but the free oxygen, but in the very small amount. You can say they cannot take the full exposure towards the oxygen, so they just need it in very small quantity. Like some minerals, some nutrient we need in a very small quantity inside our body. We need them, but if the amount will be increased inside the body, they will be behaving like the toxins or toxicity increased inside our body. So same with the microaerophils, they can grow best if the presence of oxygen are very small. But if the amount of oxygen will be exceeded, they can be, they can stop their growth, and sometimes that exceeded amount of oxygen can kill them. Canophiles, so as the name suggesting, they are carbon dioxide loving organisms that thrive under the condition of low oxygen. So canophiles basically they grow under the carbon dioxide situation. That as we need oxygen for survival, they need carbon dioxide for survival, and sometimes very low amount of oxygen also for do some specific kind of function inside them. But most of their function, most of their survival, that is based on the carbon dioxide. Facultative anaerobes, so they carry on anaerobic metabolism when oxygen is present, but shift to anaerobic metabolism when oxygen is absent. So this is condition based. Like they need oxygen for some of their process, but when oxygen is not present there, they will not going to die. They will take a alternative to further their survival. So just if the oxygen is present, they will continue their metabolic activity by having the aerobic metabolism. If oxygen, for some reasons, if oxygen will be not there, or they will be present in the situation where oxygen is not present, then they will shift to the anaerobic metabolism. So they are doing this just for their survival. So that's why facultative anaerobes can be used widely inside the industrial uses or brew industry. So we can easily make them tentable for both the process, aerobic process also and anaerobic process also. Aerotolerant anaerobes, so they can survive in the presence of oxygen but do not use it in their metabolism. So, they do not have any effect of oxygen on their body at all. Means they are not going to use it, but they are just neutral for the oxygen. Neither they are going to use it, and nor the oxygen going to effect on them have in a bad situation or bad condition. So actually, they just take it like oxygen is present or not, they don't have any effect on them. Obligate. So obligate means oxygen must have a specified environmental condition, like the obligate anaerobes. They must have free oxygen, but that is a condition there that they needed it to be free. They cannot be like if the bandage situation, they cannot grow inside them. Speculative means they can adjust or tolerate environmental condition, and they can even change or alternate their tradition or alternate their function to for their survival. Like organism is able to adjust to and tolerate environmental condition, but can also live in other conditions. So they are just you can say they are the one who can survive any kind of problem or any kind of environmental situation, but obligate, they are specified to some certain kind of situations. And you can see the pattern of oxygen use. Obligate aerobes, they will be grow like this here. Speculative anaerobes, condition can be mold. Some just go for the condition based. Obligate anaerobe, they are growing at the bottom because at this place they do not going to have oxygen. Air tolerant anaerobe, that is also can be present because oxygen is not going to affect us on any basis. Microaerophils, so they need oxygen but in a very little amount. So that's why they will be presenting in a median section. So in this median section, they are not going to have much oxygen. They're just going to get the very small amount of oxygen what they need for their survival. Now, the fourth point is here the condition that is called the hydrostatic pressure. So, what happens inside the hydrostatic pressure? So, water in oceans and lakes exert pressure and exerted by standing water in a proportion of its depth, as the name suggesting hydro and static. So, hydro means water, static when the condition is a stable condition pressure. So water in oceans and lakes. So basically in oceans and lakes, we assume that the water is not in a running position. they are in their static portion. So lax exert, they will going to exert pressure in the things which are present inside them. Water always have a pressure. So the pressure which purport on the particular organism which are present inside them. So on the basis of their pressure, some microorganism can tolerate that pressure, but some cannot tolerate that pressure. And on the basis of their tolerance, they will be presenting inside ocean, lakes, or the running water. So pressure doubles with every 10-meter increase in depth. So as well as we are going inside the very depth, or you can say the downward in the ocean or lake, the pressure will be increasing like the double, like 10 meter. If we are having the 10 atm pressure, then more we are going the downside, the double will going to face the hydrostatic pressure. So here a term which are specified for the bacteria who grow under the hydrostatic pressure. So the barophils. This is a term. Bacteria that live at a very high pressure but die if left in laboratory at standard atmospheric pressure. So they are just very, they are just toleratable or not even toleratable. That is a very natural situation for them. Barophils, they just can live. That is normal situation to having high pressure for their group. But if we put them into the normal or standard atmospheric pressure, they will be going to die. So this is a situation because their whole pattern, their whole body has been, designed to live inside a very high pressure. They cannot. Low pressure would be a very bad, you can say, unfavorable condition for their growth. So that's why the barophiles are the bacteria which you have to remember, just like they can live inside the very high pressure or hydrostatic pressure, or if we are going to put them into the normal kind or standard atmospheric pressure, they will going to die. Osmotic pressure. So the environment have osmotic pressure to us. So environment that contain dissolved substances exert osmotic pressure, and pressure can exceed the exerted by dissolved substance in the cell. So when we dissolve something inside, like we have a solution, we dissolve some microorganism inside that solution. So basically, osmotic pressure that is in comparison to their environmental pressure work on them. So there is two condition on the basis of osmotic pressure in the microbial growth. So what are these two conditions? One is hyposomatic environment, and second one is hyposomatic environment, hyper and hypo. So what happened in the hypersomatic? This is like the plasmolysis, means cell loses water and undergo the plasmolysis, shrinking of the cell. So when these kind of bacteria which do not tolerate this kind of pressure inside the water, they will going to lose the water and undergo the plasmolysis, or you can say the shrinking of cell. Hypostomatic cell gain water to maintain the pressure. What they will going to do? They going to gain the water and swell and burst. But they think that they can maintain by having the water inside them, but when they doing absorbing the water inside their body, but they are not going to attain the optimum temperature, optimum osmotic pressure, and by swelling them excessive of water inside them, they are going to burst themselves. So this was the concept of plasmolysis. You can see in a diagram. So this is a plasma membrane and the cytoplasm. If what we have normal isotonic solution, they are going to take that inside. Under these conditions, the solute concentration in the cell is equivalent to solute concentration of 0.85 NaCl. So that if this is balanced, then there will be no absorption and no exertion. But if the concentration, or if the solution is the hypertonic, so the concentration of solute like NaCl or sodium chloride is higher in the surrounding medium than the cell. So water leads to leave the cell, road to the cell is behind. So if concentration is high in the surrounding medium, I think all about how the plasmolysis work. So if the concentration higher in the surrounding medium, water have to be grow have to go from the inner side to the outer side to maintain the balance. And in order to maintaining the balance, what he will going to face, he will going to face the plasmolysis for in result of shrinking of the cell because of excessive loss of water. Halophiles, also a condition of the bacterial growth, like the salt-loving organism. I already told you what are the meaning of halophiles. So they are the organism or they are the bacteria which can live only to the optimum value of the quantity of salt, or you can say the sodium chloride. So membrane transport system, or you can say their membrane transport system actively transports sodium ions out of the cell and concentrate potassium ions inside. So by having this mechanism, they survive their whole metabolic activity, just actively transport sodium ions out of the cell and take up the potassium ions inside them. So this is a major procedure even the hydrochemical gradient when developed from inside the membrane of mitochondria also. So or in the nervous system when the action potential generates. So these sodium ions and potassium ions have a major role to play. So that's why then halophile conditions also these ions can regulate the metabolic activity of microbes. Why do halophiles require sodium? So basically, cell needs sodium ion to maintain a high intracellular potassium concentration for enzyme function. So all their enzyme function inside the cell of microorganism, what they need a very high intracellular potassium concentration. And how they are going to gain high potassium concentration? By releasing the sodium. And this release of the sodium can be done only when they are going to live in a halophile situation. Then only they are having more than optimization, or you can say more than optimum level of the ions presence. Cell needs sodium to maintain the integrity of their cell wall, and also sodium is required to maintain the you can say integrity or the way they were before. So the in the same manner, so neither they be destroyed for some any kind of other reason. So that's why they have to be integrate their integrity set. So sodium ions are going to help the microbial diversity and to maintain their cell wall. Response to cell on the basis of like halophile, moderate halophile, and extreme halophile. So they are the concentration of the salt on which these microorganisms are growing. Like nanohalophiles required a slow salt concentration. Moderate halophiles they will be need a moderate thing, neither high salt concentration and neither nor the low salt concentration. Extremophiles they will going to need a very high amount of the salt concentration for their growth. This is at the Great Salt Lake in Utah. I think you heard about the Lake of Utah. They yearly they like you know by this lake only the whole of the amount of the salt can be made is like 4.5 billion or tons of the salt can be made just by this single lake. So you can think how salty this lake it is. Most of the nothing can be grow inside this lake. No fish can be grow because the very high salinity. I think they have the salinity is approx like 15% of the saline. Normal salt concentration that can be tolerated is in the oceans. That concentration is like 4 to 5%. But in this lake, the concentration level, or you can say the concentration of salt is like 15%, twice of the oceans. So that's why, and they do not have as much big as the oceans are, but still the concentration of this salt, or you can say concentration of the solute or salinity or of this lake make very valuable for some of the industrial purpose or getting the ions or solute from it. Okay, so this is all for today about like we already completed all the physical aspect of growth inside the microorganism, and later on we're going to see about the chemical parameters. Okay, so now we will going to see some chemical requirement, or you can say as we see that there are some physical requirement for their growth. So with the physical requirement, these nutrients they also need some chemical things or chemical factors which are going to help the microorganism to grow. So, what are these chemical requirements? Carbon sources, nitrogen sources, sulfur and phosphorus, trace element like copper, iron, zinc, and cobalt, and vitamins. So these are very, you can say that these are the things which are required for their biochemical activity. Those were the things which are required for their physical aspects, but this is the thing which are going to affect how they biochemically react. So first requirement we are seeing the carbon. Why we need carbon inside the microorganism? So carbon needed here for the structural organic molecule energy source. So basically that is a structural organic molecule. It helps them to meet their structure. Energy sources, they also provide the energy there. Chemoheterotrophs use organic carbon source. So basically, some of the or not all the bacteria they are autotrophs or they can make their own food. So what happened in this case or in this condition? These bacteria are need some chemicals to by breaking down these chemicals they are going to use them as their organic carbon source or food source. So that's why the chemoheterotrophs they uses this organic carbon as their food source or nutrition source. Autotrophs, they use carbon dioxide. Okay, so in the case of heterotrophs, they just make it as their food. And if we are talking about some autotrophs or the thing or microorganism which can make their own food, so basically in that phenomena or photosynthesis, what they are going to need, they are going to need the carbon dioxide. Second chemical requirement is nitrogen. So in amino acid and proteins, so you know that this nitrogen, this is a building block or in between the amino acids and protein. So that's why we need nitrogen as a fundamental chemical requirement for microbiological growth. Most bacteria decompose protein. So most of the bacteria their function as a decomposer what they are going to decompose they are going to decompose the protein molecule. So when they are going to decompose the protein molecule the amino acids will be formed. An amino acid's basic component is nitrogen, so that's why nitrogen has to be present there. Some bacteria use NH4 or NH3 means nitrogen fixation. So some bacteria they can use nitrogen fixation for their survival because as a byproduct of nitrogen fixation there are many things which they are going to use in their cycle. A few bacteria use nitrogen in nitrogen fixation and some can be used these inside the nitrogen fixation nitrate and the nitrite products which are the intermediate situation inside the nitrogen fixation. So we can say that the nitrogen is like the elementary thing or like as the carbon they needed so same they are going to need the nitrogen for their growth and solidity. Chemical requirement of sulfur. So in amino acids, thymine and biotin group. Okay, if sulfur is not present, they cannot make the thymine and biotin, which is require the sulfur presence. Most bacteria decompose protein. So as they are decomposing the protein, they need the bacteria which going to use sulfur oxide or hydrosulfur. So that particular for that particular purpose, they also need this sulfur molecule or sulfur gas. Phosphorus in DNA and RNA and ATPs. So you know that phosphorus ribophosphate we run it deoxyribophosphate DNA ribonucleotide acid RNA. So this DNA and RNA they have the building blocks of phosphate group. So this ribophosphate that is a kind of thing which are required inside the formation of DNA and RNA, and also the ATP. ATP, you know, it is the currency of the body. If there is no ATP inside the body, then there will be no function that is going to perform in there. So for performing the several kind of functions inside the body, what we need the ATP. So that's why in ATP, what is ATP? It's adenine triphosphate. So they are involving the three component of the phosphate. So that's why phosphate is a fundamental unit and fundamental element or chemical which are required inside the biochemistry of microorganism. And membrane also need cell wall membrane. They also need phosphorus. Phosphorus is a source of phosphorus. So this PO4, that is also a source for the phosphorus formation inside them, and this by which they can get the phosphorus for their survival. Trace element. This is another kind of a chemical requirement. Trace element means some inorganic elements, some minerals which are required in a very small amount. Like in the plant, in our body, for our survival, we also need two kind of element. Some element which we need in a higher amount, like the macro elements, but some we need in a very tiny amount, and that are called the micro element. So basically, this trace element, they are like the micro elements or micro molecules which are required, but required in a very small quantity, usually as enzyme cofactor like iron, molybdenum, zinc. So why we are needing him here for working for enzyme cofactor? Because sometimes enzyme cannot do their function independently. They need some cofactors working along them. So at that time, these trace elements like iron, molybdenum, zinc, they are working for these trace element and helping them to form a better reaction or to do a better reaction. Third is here buffer. So to neutralize acid and maintain proper pH. So basically neutralizing, why we use buffer? We use buffer to neutralize something. And what we are neutralizing here? We are neutralizing here the proper chemical environment of the microorganism growth, and we are giving them a proper pH. Peptone and amino acid or phosphate salt may act as a buffer. So peptone or amino acids, they can also work as a, or you can say they can also as a used as a buffer, or they can maintain the acidity or the alkalinity, or you can simply can say the pH of the environment, and at their optimum pH, microorganism can grow in a favorable way. Organic growth factors, they are also responsible for the growth of particular kind of microorganism, like organic compound obtained directly from the environment, vitamins, amino acid, purines, and pyridine. So they are the thing which we are need, or sorry, microorganisms are need for or from the environment to grow in a better way, and they are their growth factors. Without these, they cannot grow in appropriate manner, because they are the key for several metabolic activities inside them. So for doing their metabolic activities, without interpretation, they will going to need these things. Now we will going to see preparation of culture media in our next lecture. So this is all about today. Like we already see that how this microorganism needs some physical factors and they need some chemical factor for their survival and for their growth. So this is important, and these are the key points you have to remember. The things which are required for the nutritional growth, some physical and some chemical factors. So in some physical requirement, we have temperature, pH, oxygen, hydrostatic pressure, and osmotic pressure. And for the chemical requirement, we need carbon, nitrogen, sulfur, phosphorus, trace cement, oxygen, organic growth factors. So the major motive was of this lecture was to tell you to you have to know what are the requirements of for the growth of microorganism, and the requirement are two types: physical requirement and chemical requirement. And on the basis of having optimum amount of their requirement, even we are talking about physical or chemical, they can do the growth at the optimum level. So in the next lecture, we are going to see how these growth factor will going to help us inside the culture media preparation, and we are going to make culture medias to grow the microorganism at our lab level. This is all for today.